Okay, so transcription. I put Roman numeral headings for transcription number 1 and then Roman number 2 for translation. Sometimes because they start similarly, it can be easy to flip-flop, but so hopefully this helps. This is going to come first, transcription. You're taking DNA to make RNA. So one of the 2 DNA strands is going to be called the template strand. So if you remember, DNA is a double-stranded helix, so there's 2 strands that are going anti-parallel to each other. One of them, say it's this top one, is called the template strand because it's going to be like read as like a template, and then based on what bases are there, what nucleotides, that helps to build the new RNA molecule. So one of the 2 strands is the template strand, and it provides a template for ordering the sequence of complementary nucleotides in an RNA transcript. So remember complementarity, like G and C always go together, C and G. So based on what nucleotides there is what is going to be put on the RNA version. And so the template strand is always going to be the same strand for a given gene because that recipe is always going to be the same, right? But it's super interesting and complex, but interesting that, however further along the chromosome, the opposite strand might actually be the template strand for a different gene. So it's super interesting. It makes it more complex for us to learn about, but it's really interesting and like efficient. And so specific DNA sequences associated with the gene direct which strand is going to be used as the template. So there's going to be some sequence that says, okay, this is going to be the template for this recipe. And the mRNA molecule produced is complementary to the template strand. And then this point will make a little bit more sense later. But so when you build the mRNA molecule, there's also going to be that directionality of like 5 to 3. And so during translation, which we'll talk about more later, those mRNA nucleotide bases are going to be read in groups of 3 or triplets. And then so one group of 3 bases is called a codon. And so those are going to be read in the 5 to 3' direction. So then the non-template strand is called the coding strand, because if you end up comparing the nucleotides to the mRNA that you made, it's basically almost the same sequence. It's just like DNA version instead of the RNA version. So that's why the non-template strand is also called the coding strand, because that's basically the code. Because the nucleotides of this strand are identical to the codons, except that T is present in the DNA instead of the U in the RNA. So there's, not identical, but basically. And then so later on, each codon specifies the amino acid that is going to be placed at that position along a building polypeptide. So that will make more sense later when we talk about it, but just for some reference. And so here I made one of those worksheets. I hope they help you. And so this is representing transcription. So you take one DNA molecule and you unzip it in a specific region. And then so one strand is going to be read, and then so this, like as in it's going to be read to write something. So this is going to be the template strand, the one up top. Template strand, okay. And so this is what's you're gonna look at what base is here, and that'll dictate what you're gonna put here in the mRNA that you're writing. And that other strand is gonna be the coding strand. And so this worksheet is like a super overview just to give you the big picture, and then you could fit in smaller details. So like we're doing now, kind of. Coding strand. But I always say that it's better to have the overview and then fit in the details. And then so what you're building here is RNA, right? And more specifically, it's pre-mRNA. So it does get altered later on, and then the final version is mRNA. So this is RNA, and so what you do is you would read what letter is here on the template strand and put the corresponding complementary letter. So remember, in DNA, A and T always go together. And so if this is a T, then this is going to be an A. But in RNA, instead of a T, RNA uses uracil instead. So this is going to be a U. But otherwise, it's the same. If there's a C, you're going to put a G. If there's a G, you're going to put a C. If there's a T, you'll put an A. But yeah, if there's an A, you will put a U. So remember, that's a big difference. So in DNA, you might want to say it like that. DNA. So A would be with a U in RNA. T is always going to be an A. G is always going to bind with C, and then C is always going to bind with G. So that's the main difference, is that in RNA, it's going to be a U instead of a T. Okay, so if this is a C, you would put a G. If this is a C, you would also put a G. G, C, A, in this case, instead of a T, it's a U, because again, this is RNA. G, C, G, this is a C, C, this is a G, A, it's a U, and then these are both going to be A's. Boom! And so that is how you build RNA. And so the word transcribe basically means to write. Like, it's to write something based off of a copy or based off of something. So like there are people in court who are like the stenographers, like they're listening and then they're writing something down. They're kind of transcribing stuff. And then you read the transcription. Or if you look at it like a video recording, you could, on like YouTube or whatever, you can look at the transcript based on what they said. It writes down something, right? So, and then also back in the day before there were like machines to copy books, there was a guy who would like read this book and then transcribe a new copy of that book based on that original. So transcription, you're reading something, in this case DNA, and then you're writing something, in this case RNA. And so I hope that you do this practice. You know, if you don't print the worksheet, then copy it down or something. I think it helps to really like do that for yourself. Okay, and then so we'll kind of come back to the rest of this in a little bit. But that's like the overview, you know, DNA double-stranded helix gets unzipped. One strand is the template that you read to put what's there. And then the other strand is the coding strand, because if you look, it's basically the same sequence if you do it right. Okay, so now to add some more detail. So basically what you were doing was the job of RNA polymerase. So RNA synthesis, or building RNA, is catalyzed by the enzyme RNA polymerase. You're making a polymer of RNA. And so this pries the strands apart and joins together the RNA nucleotides. So if you wanted to, on that worksheet, you could then add in like a blob to show RNA polymerase. And so the RNA is complementary to that DNA strand because based on what like matches up. RNA polymerase does not need a primer. So as opposed to, remember, DNA replication, there is DNA polymerase who's building a DNA molecule. but it needed to have an RNA primer. But in this case, for whatever reason, it just evolved that RNA polymerase does not need a primer. It already, was made out of RNA. And so RNA synthesis follows the same base pairing rules as DNA, except that uracil substitutes for thymine. And so I, gave the big picture, but there are 3 stages. So stage 1 is initiation. So you're kind of just starting. Stage 2 is elongation. So as you're building the RNA, it's like growing. And then stage 3 is termination. So you're stopping the transcription. You don't go on forever. You only want to copy down this one recipe. So we will break that down. So initiation of transcription. So there are different regions that are going to be called different names. And so this is going to be some more terminology that you're going to have to kind of memorize, maybe make flashcards for. So there is a sequence of DNA around a gene called a promoter. And so even though this has a name, this is just a DNA sequence. It's like a little segment of DNA. And so the promoter is the DNA sequence where RNA polymerase attaches. So remember, RNA polymerase is the enzyme whose job it is to build this, and so it has to kind of start and attach somewhere. The terminator is a sequence in bacteria. So in bacteria, the terminator is a sequence signaling the end of transcription. So this is like, okay, this is the end. You can stop writing this recipe now. But that is specifically in bacteria. Promoters can be in either one. And then that stretch of DNA that's transcribed is called that transcription unit. So promoters, again, the sequence of DNA, help signal the transcription start point and usually extend a little bit upstream of the start point. So if we think again about the analogy of like your genome is one big recipe book, you're looking for the title of that recipe that you're looking for. So say you want to make like a German chocolate cake or whatever. You're looking for the title that says German chocolate cake, and that's where you're going to start making your like notes, right? So the promoter signals like, okay, this is the start of this recipe. This is where you're going to start transcribing. And usually it's just like a little bit ahead of the actual start of the recipe. So kind of like the title, it's a little bit ahead of the actual recipe, but it's kind of important. That tells you where to start. And so in eukaryotic cells, there are other things that help RNA polymerase. So in eukaryotic cells, there are proteins called transcription factors that help guide the binding of RNA polymerase and help initiate transcription. And so when those transcription factors come together, and then RNA polymerase come together, they make what's called the transcription initiation complex. It's kind of like when all the Power Rangers come together and make that one huge Power Ranger. Like once they're all together, they can like start doing the job and whatever. So this whole complex is the completed assembly of transcription factors, these proteins, and RNA polymerase bound to that sequence called the promoter. So again, that's kind of like the title of the recipe, like this is where we're going to start, and then we're going to go. And there is a sequence, because again, it's just a sequence of nucleotides. There is a part of it that's called the Tata box, and that is really important in forming this whole complex in eukaryotes. So here's a picture. So if this is all just DNA, this is representing, these 2 lines are representing the DNA double-stranded helix, So this stretch of sequences is called the promoter. And then there is a part of the promoter that's called the Tata box, and it's called that because literally it says TA. And then there's 3 more A's on one of the strands. And so those are necessary. So the promoter is kind of, again, like the title of the recipe. And so that's where RNA polymerase and these transcription factor proteins come together and assemble into this whole complex, and then they can start transcribing. Yeah, and so that is like the official start point, but this is kind of signaling this is where you get started, and then start now. And then so it starts transcribing. And remember, just like how DNA is always going to grow 5 to 3 prime, you're always going to add the new nucleotides at the 3 prime end, with the hydroxyl group sticking out. It's the same for RNA. So it's always going to grow like that. Like the 3 prime end is always going to be the growing end. Which I'm getting ahead of myself because that is elongation. So as RNA polymerase moves along the DNA, it untwists the double helix 10 to 20 nucleotides at a time, so it can like read them and then put down the nucleotides. Nucleotides are added at the 3 prime end of that growing molecule because just that's how it has to happen at the chemical, molecular level. That's how you add them. And transcription can go at a rate of 40 nucleotides per second in eukaryotes. And so one gene can actually be transcribed simultaneously by several RNA polymerases. So say it's like an important gene for an important enzyme that you need a lot of. So that gene can be transcribed by a bunch of RNA polymerases at the same time because they're like, we need this recipe now. Let's have 5 different chefs making this recipe because we need it. And so this is elongation. So yeah, this whole kind of like white bubble looking, arrow looking thing is DNA polymerase. It unzips a few, 10 to 20 nucleotides at a time in the DNA, and then it reads the template strand and then starts building the mRNA. And so remember those like anti-parallel rules. So the template strand is going to be going 3 to 5 prime because it has to build the new RNA 5 to 3 prime. It has to add the nucleotides at the 3 prime end. Yeah, and then the base complementary rules. So if there's a T, you put an A. If there's an A, you're putting a U because this is RNA, and so on. And then so transcription, termination. So the mechanisms are different in bacteria and eukaryotes for like when to stop transcription. So in bacteria, the polymerase stops transcription at the end of the terminator sequence. So remember, the terminator is just like, this is where you stop. But that's only in bacteria. So the polymerase stops at the end of the terminator sequence, and then the mRNA can then be translated without really any changes, any further modification. But in eukaryotes, there's a little bit of a difference. So in eukaryotes, RNA polymerase, and there are actually different ones, but it's specifically RNA polymerase 2. So RNA polymerase transcribes this sequence at the end that's called the polyadenylation signal sequence. So it's like a little bit of extra stuff at the end. And so it transcribes that little bit of extra stuff, the polyadenylation signal sequence, but it is important. So it has to go a little bit past the gene and include that sequence. Then the RNA transcript is released, 10 to 35 nucleotides past that sequence. So there's like a little bit extra added on in the eukaryotic mRNA. And so this is an overall summary of transcription. Even though there was a lot of detail, big picture, there is a promoter sequence, that's the DNA, where RNA polymerase and the transcription factors are going to meet up and then start working. This is the RNA that gets built from 5 to 3 prime. And then that one recipe that gets transcribed is the transcription unit. And yeah, like say the gene ends around here, then like this much might be the polyadenylation sequence. So that's the end of transcription, but that's the first draft mRNA, so the pre-mRNA. So enzymes in the eukaryotic nucleus modify this pre-mRNA before the genetic messages are sent to the cytoplasm. So that's why it's pre-mRNA. So during this process of RNA processing, both ends of the primary transcript are altered. And additionally, in most cases, there are certain interior parts of the molecule that are cut out and then kind of the remaining parts spliced together. So each end of the pre-mRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide 5' cap. And then on the 3' end, it gets a poly A tail. So here's a picture. So this is the 5' cap. It goes on the 5' end, and it's a modified guanine nucleotide. And then at the other end, at the 3' end, there's a poly A tail. It's really just a bunch of adenine nucleotides, 50 to 250. Yeah. So what is the point? What do these things do? So these modifications share several functions. They seem to facilitate the export of mRNA to the cytoplasm. They protect the mRNA from hydrolytic enzymes that are in the cytoplasm. So remember, hydrolysis is when you like break apart molecules. So having these things on the end kind of help protect the mRNA from these enzymes just breaking it down immediately. And then they also help ribosomes attach to the 5' end for translation. So they help by adding these little things. And so you could see this figure shows that polyadenylation signal. There's like a lot of adenines again, and so that is important. And so most eukaryotic genes and their RNA transcripts actually have long non-coding stretches of nucleotides. So there's like a bunch of nucleotides, but they don't really code for anything. Like it's not part of the final product of the polypeptide and the protein. It's kind of just like fluff. And so sometimes those get cut out. So most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides that lie between the coding regions that actually code for part of the polypeptide. So these can be removed through what's called RNA splicing. So back in the day when films used to be actually like recorded on film, and you would kind of splice different scenes together, you would have to cut that like film and then basically glue and like merge it, like this totally different scene with this totally different scene. You cut it, cut out whatever's in between, and then like merge it and splice it together. And so yeah, these introns, these segments get cut out and then spliced together. So the names for these different regions are kind of inverse, it feels like. But so introns are what are going to get cut out. Introns are the non-coding segments of a gene, just like the fluff. And these are intervening sequences. So that's why I think they're called introns, intervening. Or that's how you can remember it. Because it's just like the intervening parts between what really matters. So the introns are what get cut out. The exons are the regions that are eventually expressed. So you can remember it that way as well. Exons get expressed. So those are the ones that get to stay and then get spliced together. These usually get translated into amino acid sequences. So here you can see this is a pre-mRNA on top. It already has the 5' cap and the poly A tail. And so there are segments that are like different colors of orange. So there's like a darker orange and a lighter orange. The lighter orange is showing introns. So these are the segments that don't code for anything that's going to be in the final product. So these are going to get cut out because these are just intervening sequences between like the important stuff. So the exons are in the dark orange. So you cut out the introns, and then what you're left with are all the exons spliced together. So even though this pre-mRNA might have been like this long, it's going to be shorter in the final version, and then it's just called mRNA, messenger RNA, or sometimes it's called the mature mRNA. And so how does this actually get spliced? So the removal of introns is accomplished by these like structures called spliceosomes. So spliceosomes have like a variety of proteins and small RNAs that recognize those splice sites. So it's like a complex of proteins and small RNAs. They recognize these splice sites that are saying like, hey, this is an intron, this is just fluff. And then so that is what also like the RNA of the spliceosome also catalyzed the splicing reaction. And so here is a picture, so a whole spliceosome, and there are little small RNAs inside that are part of it that recognize parts of the intron. And they help say like, okay, we got to cut all this stuff out. And so they cut out from here to here based on what those small RNAs recognize inside of a spliceosome. It gets formed into like a loop, and then that whole thing gets cut out. And then the exons get spliced together and the exons get expressed. So it's another case where the terms can be easy to flip-flop, but try to remember like exons are what are going to get expressed. And then there are also ribozymes. So ribozymes are catalytic RNA molecules that function as enzymes. So that's kind of why it ends like zyme, and can splice RNA. So there are 3 properties of RNA that are really interesting that it lets it function as an enzyme. Because RNA can form 3D structures, because how RNA can like, it's single-stranded, but it can meet and base pair with itself. Some bases in RNA have functional groups that can participate in catalysis. And then RNA can hydrogen bond with other nucleic acid molecules. So that's how like, why are RNAs part of these things is because RNA is able to like do these things that kind of have it function like an enzyme. Okay. And so some introns, not all of them are just like useless. Like maybe it doesn't code for something, but sometimes there is a purpose. So some introns have sequences that regulate gene expression and may affect gene products. Some genes can code for more than one kind of polypeptide, depending on which of those segments are like included and cut and pasted together. So you can get different exons and cut and paste them together in different ways. And so this process is called alternative RNA splicing. And so in this way, like from one recipe, you can make a bunch of different kind of sub-recipes. So the number of different proteins an organism can make is actually much greater than the number of its genes, which is again, super efficient. You have one gene, but if you can make like 5 different proteins from this, it's just very efficient. In a way, it's like if you do read a recipe online or something, for example, and then online, a lot of times they'll have like little paragraphs of like extra information, like, oh, if you like this recipe for lasagna, even though this is a meat lasagna, you can maybe take out this and it'll be a vegetarian lasagna. If you wanted to make it a vegan lasagna, you could substitute this for this. If you don't like, if you have high blood pressure, you could take out the saltiness of whatever. So you know how, yeah, some recipes have extra information, but it's not all useless. You can kind of pick and choose what you want to include in your recipe. Think of that like introns. Introns have sequences that can help change the final recipe. based on what you include or don't include. So again, proteins are actually large and have different polypeptides. And proteins often have a modular architecture consisting of discrete regions called domains. So in many cases, the different exons will code for a different domain in one protein or one polypeptide. And then we'll show a picture in a second. So if this is a polypeptide, there are different domains. So the different exons can code for different domains. And then if you cut and paste different exons, you have different domains and ultimately a different protein. So exon shuffling can result in the evolution of new proteins by mixing and matching exons between different genes. So here, if a gene has exon 1, some fluff, some intron, exon 2, another intron, and then exon 3, if you express these 3 exons, you have this polypeptide with these 3 domains. But you might even get away with, like, maybe you want to make a different version, and so you only express and cut out and express exon 1 and 2, then you make a different polypeptide that's only this. Or if you wanted to make exon 2 and 3, then you would make this polypeptide. So yeah, each exon can code for a different domain of a polypeptide. And so this is the overall summary of RNA processing. You go from a pre-mRNA or a primary transcript, kind of like the rough draft. You add on a 5' cap and a poly A tail, and then you cut out the introns, and then you splice together the exons to be expressed, and that's your mRNA. And so that is what I tried to represent back in the worksheet. So that's the pre-mRNA, and then this is the mRNA. It's the 5' cap. The poly A tail. And so basically through transcription, you make one mRNA. And then I would rewrite this, but I don't want to take up that time. And then maybe future versions of this worksheet, I will cut out some stuff, and then that way you could see splicing. But yeah, so that's supposed to be like represent RNA processing. And so that is transcription. Yes, and then next would be translation.